Hi everyone, I am Dr. Sheela, Assistant Professor, PG and Research Department of Microbiology at St.Joseph Arts and Science College at NMS Cuddalore. Now I discuss about the topic DNA replication. The process of synthesizing the new double helical DNA from the existing DNA is called as replication. Replication produces identical daughter molecules of DNA. Model of replication. Their model of replication had been proposed. They are conservative replication, semi-conservative replication, dispersive replication. Conservative replication, according to this model, the parental DNA is conserved to one daughter cell and the newly synthesized DNA to another daughter cell. This method was proposed by Cavalieri. Dispersive method. According to this model, the parental DNA is unequally distributed to the daughter cell. Next one is semi-conservative replication of double-stranded DNA. In this semi-conservative mode of replication, each parental DNA strand serves as a template for new strand. The new strand is connected with the parent strand by hydrogen bond. During the replication, the parental double helix unwind and then rewind again into two new double helix. Each double-stranded DNA contained one parent strand and another one is new strand. That is semi-conservative replication of double-stranded DNA was proved by as an experiment of Matthew Milson and a Franklin Stahl experiment. This one is Michelson-Stahl experiment. In this experiment was isolated before the shifting the low density medium and sedimented to the equilibrium cesium chloride. The density of DNA was approved 1.722 gram. The same culture also cultivated in the medium, nitrogen and the DNA extracted in cesium chloride and the weight of 1.708. The results indicated the one strand from nitrogen 15 and another one is nitrogen 14. In a second strand of replication in nitrogen 14 medium and first DNA converted into equal amount of two species. So, in this experiment proved the semi-conservative replication of DNA. The geometry of DNA replication. Unwinding double helix, a semi-conservative replication, is an important mechanical problem. If the DNA molecules were fully extended in a solution, there would be no problem. But it's difficult to maintain such a condition. For example, the DNA E. coli 600 times longer than the cell. A simple solution is forming and maintaining break at one parental strain ahead a growing fork. That the break is performed before the replication forks is past the nick. Otherwise, the daughter strand would be lost. The double-stranded DNA is twisted or coiled 360 degrees for every basis. The prokaryotic DNA complex, because they circular and no free end, the DNA is also usually highly folded into compact structure. The structure of DNA during replication resembles the Greek letter theta, so it is called as theta replication. As the replication is the two daughter strand proceeds along to this double-stranded DNA helix. The non-growing end of the daughter strand cause the unreplicated DNA become unwound or tight sometimes. This may result in the positive supercoil of unreplicated portion. The separating the tightly twisted DNA is accomplished through topoisomerase enzymes. These enzymes making just ahead of replication of DNA. Basic requirements of DNA synthesis: substrate, deoxynucleotide triphosphate are the substrate for DNA synthesis. The four types of deoxyribonucleoside, triphosphate and adenine triphosphate and deoxy GTB and DCTB and TTCP. Next one is template. Each of the parent strand act as a template for the synthesis of new DNA. The template guides the addition of appropriate complementary nucleotide to the newly synthesized DNA strand according to the Watson-Crick base pairing. Third one is primer. A primer is a segment of short nucleotide which is the complementary to the template with a free of 3' hydroxyl group to which a nucleotide can be added so that the nucleotide are polymerized during this DNA synthesis 5' to 3' direction. Most of the primers are RNA primer. Fourth one is proteins and enzymes. Many enzymatic and non-enzymatic proteins are required for translation. They are polymerases, helicases, topoisomerases, ligases, and the SSB protein. Next one is DNA polymerase. The DNA polymerase Arthur Kohrenberg in 1955 characterized the DNA polymerase from E. coli cells. Later, E. coli cells were found to contain 5 types of polymerases. They are polymerase 1, polymerase 2, polymerase 3, polymerase 4 and polymerase 5. The polymerase 1 has two fragments. The removal of wrong paired nucleotide added into the replication. The activity is called as proofreading. Next one is polymerase 2. It is involved in DNA repair. Next one is polymerase 3 is principal replication of enzymes of E. coli. Next one is E. coli polymerase 4 and 5. They involved in unusual repair. Next one is helicases. The separation of two parental DNA strands is carried out by the enzymes helicases. That is DNA B protein. Third one is topoisomerases. The strand separation by helicases created topological stress in the helical DNA, which is relieved by the action of topoisomerase. Fourth one, primases. Primases are short segment of nucleotide, generally of RNA, and initiate the replication. These are synthesized by primases. Fifth one is DNA ligase. It is a mix of polymerizing DNA backbone or sealed by the DNA ligase. The nicks in Ogasaki fragment of lagging strand. The nick on circular DNA after leading strand synthesis and the nick after primer remove all the sealed by ligases. Sixth one is single stranded binding protein. This protein maintains the DNA unpaired form. The SSB protein are stripped of the DNA just before it can be replicated by polymerases. Next one is mechanism of replication. The prokaryote DNA replication in different mechanism like a theta replication, the theta replication rolling circle model and a replication looped rolling circle model of replication. The E. coli chromosome replicated by the bidirectional mode of single replication origin. The DNA unit undergoing replication is called as replicon. All the protein particles associated at the site of replication as a single unit is called as replisome. The replication of DNA proceeds the 3 stages. First one is initiation, elongation and termination. Initiation, it is a replication of E. coli chromosome is initiated its origin site of replication. It's called ORIC. Origin C. The origin C site, it is a 245 base pair sequence within the 9 base pair repeats of the 3 base pair repeats play significant role in replication. About 9 protein particles participate the initiation of replication. They are DNA A, DNA B, DNA C, DNA G, SSB protein, RNA polymerase, DNA gyrase, histone-like protein. The diamethylase. This separated of two strands of DNA, origin C, and making it ready for the elongation process. This one is topoisomerase, replication, primase, ligase, DNA polymerase, helicase, single-stranded binding protein. Next one is elongation. In the elongation process of DNA strand are continuously copied with their complementary nucleotide. The elongation require continuous unbinding by DNA ligase, removal of topological stress by topoisomerase. The stabilization of single strand by SSB protein followed by polymerization of DNA strand by polymerase. The synthesis of two different strands. The strand synthesized in 5' to 3' direction is called as leading strand, and 3' to 5' direction is called as lagging strand. The two core enzymes of single DNA polymerase 3 binds one onto each strand elongation to the strands. This one is elongation process. First one is 5' to 3' leading strand, and 5' to 3' it is a lagging strand. The leading strand synthesis begins with the synthesis of short RNA primer of 10 to 50 nucleotides of the replication. Then the proceeds continuously with the addition of nucleotide by polymerase 3. The lagging strand synthesis is not continuous process. It occurs in short fragments called Okazaki fragments. The elongation always the direction of 5' to 3'. The DNA polymerase 1 removes the RNA primer and add deoxynucleotide. Finally, the two successive fragments are joined into DNA by ligase. This one is replication of DNA. This one is lagging strand, Okazaki fragments, RNA primer, DNA template, single-stranded binding protein, DNA primase, DNA helicase, DNA template. This one is leading strand and DNA polymerase 3. It is involved the replication of DNA. Next one is last one is termination process. The finally two replication fork of circular E. coli chromosome met at a region called terminus region. In this region contain 20 base pair sequences called TER sequence. A protein called terminus utilization substances bind to TER sequence. This complex terminated the replication of E. coli. Sometimes the completely replicated the two DNA circles are topologically linked. The two DNA linked topologically are called catenates. The two double-stranded DNA are separated by topoisomerase 2 and 4. The separation of two DNA helicase is catenates is called as decatenation. Decatenation means separate the two double-stranded DNA and synthesis the new double-stranded DNA. Thank you.